We're going to try to go through the plasma membrane right now. In terms of the AP curriculum, is 2.4. We're trying to figure out what exactly is the outer boundary to a cell and what are its components really. All right, so let's dive in. All right, so first off, what is a plasma membrane? So I'm going to go a little Travis Scott on you guys and let you into sicko mode, but we got an outer membrane here that differentiates the outside environment from the inside, and the way I feel about it is like a ball. Okay, and bear with me for just a second. Okay, so imagine if we didn't have this outer wrapper. Okay, what would we have really? Okay, we'd have the air out here, and we have the air in here, and there'd be nothing really to differentiate those two spaces, right? So the fact that we have this outer wrapper, this outer layer, is what gives Spalding all its game, right? So hopefully everybody understands here. You're not going to be able to play any game without this outer membrane or this outer layer that separates out the air from inside the ball from the air outside the ball. And for a cell, it works exactly the same way really. We have a layer on the outside that separates out the external environment from the internal environment, and that's really what we're talking about. Okay, so these things are selectively permeable, really big term. Everybody's going to use it, every textbook in the world, and pretty much what that means, to break it down, is there's only going to be certain things that we're going to allow in and certain things that we allow out. Okay, and so it's going to separate out the inside of the cell from the outside of the cell, and you can see here there's a couple different components I try to highlight in this picture. So you can have a situation where the outside of the cell might be acidic and the inside of the cell might be basic. Okay, and this is really important for some of our chemistry and our reactions, what we can accomplish outside of the cell versus inside of the cell, and that whole concept of compartmentalization, which happens in cells and in bodies of all organisms. Okay, so one thing I want to lay down right now, make sure everybody understands, is that you could have a cell change how it looks throughout its lifetime. You could have a cell change quite a bit. It can get more proteins like I showed here, or it can have more cholesterols embedded between the phospholipids. It's all about what that cell has to accomplish. Okay, and so not only can we vary, like within our own body, different types of tissues are going to have different cellular components comprising that cell membrane based on their function, but even within one cell over the course of its life, you could have alteration based on what that cell has to accomplish. And again, just like everything else in biology, form is going to follow function here. Whatever that thing has to do, it's going to find a way to get it done. And if it has to transport, we're going to have more proteins. If it has to be more fluid, we're going to see some changes to the components of these phospholipids, and we'll get into that a little bit later. Okay, so just to kind of drive home the point that these things are selectively permeable, I have a couple things here. We have an oxygen molecule, okay, which is a non-polar, very small molecule. Over here I have a virus, okay. Obviously we don't want that to be able to enter the cell too quickly. And then over here I have a food vacuole, okay, or excuse me, a food item. So let's take a look at this here. Oxygen is going to be able to go right through these phospholipids, okay. That's what we call simple diffusion. Again, we'll get into that a little bit more later. Okay, this guy is going to be prevented from getting across. Okay, that's hopefully how these cells are going to work. And then this food item, this large piece of chicken here, we're not going to be able to get that thing across real easily either. We're going to have to figure out another way to get that across, and that will form a food vacuole, and lysosomes will help to break that down. Again, we'll get into some of that detail later. That process is called endocytosis, and it's a way we can get large food items like this to cross the cell. But as you see there, only certain things are allowed to cross, and that has a lot to do with some of the components that we have for the cell membrane. So the first and the primary component of the cell membrane is this thing called a phospholipid. We got a central phosphorus here. This is a phosphorus containing region, I should say. It's not just phosphorus, there's other components up here in this phosphate head, and then below that phosphate head we have two fatty acids. Okay, those two fatty acids can be varied. So it's really important that you guys understand that if the fatty acid is completely loaded with hydrogens, meaning that it can't hold anymore, we call that a saturated fatty acid, and that's depicted right here. And what you'd see in the fatty acid is that it'd be nice and straight, right? And we've previously discussed like trans fats, and so I hope that everybody's clear on this point. But if you have no double bonds, the fatty acid again will be completely straight and completely full of hydrogens. This fatty acid, on the other hand, has a double bond right here, and that double bond is what's going to cause that bend. So the more double bonds you have in the fatty acids throughout your membrane, it's going to take up more space. It's going to cause the phospholipids to be more spatially separated, and we'll look at that in terms of the membrane overall. So if you have a bunch of fatty acids that have unsaturated bonds, they're unsaturated fatty acids, excuse me, as long as they have at least one double bond, they're going to be bent here. And the fact that these fatty acids are bent, it's going to prevent these phosphate regions from being real tight to each other. Down here, these things are right up next to each other. All of these fatty acids are lined up perfectly straight, and so this is going to be more solid, more rigid, more viscous. Any of those terms would be appropriate, but because they're able to be packed nice and tight, it's going to be more like a saturated fat, like butter or any animal fat that you can think of. Be more like a solid at room temperature. Over here, this is more like a vegetable oil or an unsaturated fat. You're going to have these things be very far apart from each other, and we would consider that to be more fluid. So a membrane has the ability to be manipulated just in the simple fact that it has double or single bonds and the number of them, right? So if we needed a cell to be more squishy, more fluid, we could have a whole crap load of these. If we wanted the cell to be nice and rigid, we'd have more of these in the outer membrane. All right, so these are all phospholipids, and they make up the vast majority of the cell membrane. And while I'm at it, I just wanted to reiterate the point that this cell membrane, this plasma membrane, even though we're talking about it in terms of the outside of a cell, this is also the wrapping to all of the endomembrane system and several little organelles. So this membrane, the same style of membrane, same components can be found not just on the outside of the cell, but also surrounding organelles. And these phospholipids orient themselves in what's known as a bilayer. So we have a phospholipid layer here and a phospholipid layer here. Because we have two, we use a prefix bi, and we have a phospholipid bilayer. Okay, hopefully everybody's able to recognize the double bond, some of these bent ones. Most of these I drew pretty straight, but typically you'd have several double bonds throughout. All right, so these fatty acids are going to face each other, and this middle region is going to be non-polar, and it's going to be completely hydrophobic. Okay, so this area here would allow like for small non-polar molecules to cross through, but it really is not too advantageous for polar molecules to try to cross this line to get into or out of cells. Okay. And then the head region, which is this top part here, which I depicted trying to highlight in blue, that head region is going to be completely hydrophilic. So that outside is going to allow things like water and anything that's polar to kind of penetrate. Also, you guys try to think about if I had like a big protein that was sitting up here and it had like some polar side groups. It might interact really well with these polar heads, like kind of attract, attach, form some weak bonds and things of that nature. Okay, so things that are polar are going to mix well with that outer region. Again, polar out here, non-polar in the middle. Okay, and hopefully everybody's following along okay.